As you might expect, anabolism and catabolism are intimately linked. Write in your notes that anabolism and catabolism are linked by energy coupling, and that energy coupling means that the energy generated by catabolic processes is harnessed and used by cells to perform anabolic processes. Now, in this way, these metabolic pathways kind of intersect in such a way that the energy released from the downhill reactions of catabolism can be used to drive the uphill reactions of the anabolic pathways. This transfer of energy from catabolism to anabolism is referred to as energy coupling. That's a very important concept for you to understand. So, if reactions are downhill, meaning they release their own energy, maybe you should get that in your notes, why don't they just happen spontaneously? Like the penguin asks, right here is the penguin. Like the penguin asks, why don't stable polymers like this starch molecule just spontaneously digest into their monomers? Write in your notes that it's because these organic polymers are held together by very stable covalent bonds. Because these bonds are so stable, energy is going to be required to get the process of breaking these bonds started. Now, from chemistry, do you remember what this energy is called? Hey, if you said activation energy, you'd be correct. Like we mentioned earlier, activation energy is just like a match. There needs to be a spark or some kind of friction to start the fire. Write in your notes that because large biomolecules are so stable, they must absorb energy in order to begin the process of breaking the bonds that hold them together. Also get in your notes that enzymes are also used to produce bread, they're added to detergents to clean stains, and they're used to treat leather in the same way. And because enzymes break down cellulose into simple sugars that can be fermented into alcohol, they're also a big part of producing ethanol from cellulose. And I give you those examples because they've been asked for before on an AP exam. Okay, let's briefly mention the second law of thermodynamics and get it in your notes. We mentioned entropy in the first podcast. Recall that entropy is a measure of the randomness or disorder of any system. Write in your notes that the second law of thermodynamics states that the universe tends toward disorder or higher entropy. Well, if that's true, why don't proteins, carbs, and other biomolecules break down? Get in your notes that at temperatures of the cell, molecules don't make it over the hump of activation energy. Yeah, that's the one we mentioned in the graphs you have in your notes, and the one that's shown here. So this activation energy. Most molecules don't make it over this hump of activation energy. This then creates really strange catch-22 for the cells, because a cell must be metabolically active, and heat would certainly speed up these reactions, but unfortunately could also denature proteins and kill cells. Not good. Okay, so then how do cells solve this damaging catch-22? Why, enzymes, of course. Write in your notes that enzymes are what we call biological catalysts, and that there are about 40,000 different enzymes in human cells, each controlling a different chemical reaction. Also write that enzymes increase the rate of reactions by a factor of between 106 to 1,012 times, allowing chemical reactions that make life possible to take place at normal temperatures, and that in addition to catalyzing the reactions of cells, such as respiration and photosynthesis and digestion. They may also act as motors, membrane pumps, and receptors. Now we've introduced the idea of a catalyst. Jargon is important in science. So you see that word catalyze or catalyst used many different ways, like the verb catalyzing you just wrote. Write in your notes that the process of an enzyme doing its job is referred to enzyme catalysis. C-A-T-A-L-Y-S-I-S. Now, exactly how do enzymes actually work? Okay, time to draw this graph in your notes and label it correctly. So this graph right here needs to go in your notes. Also write that enzymes act to reduce the amount of activation energy necessary to start a reaction, as you can see right here in this graph. So what we mean now is you can see this hump that you've written already in your notes. This is the uncatalyzed, or reaction that doesn't have an enzyme associated with it. Now take a look at this dip now in activation energy. So it's this reduction in activation energy that we're talking about now, that catalysts produce. Now, as a result of this reduction in activation energy, large molecules can now begin the process of hydrolyzing into their monomers without breaking apart or denaturing. If you don't have the word denaturing equals breaking apart in your notes, now's a good time to put it in there, because we're going to use it quite often. So make sure you get that in your notes right now if it's not in there already. Okay, so the bottom line here. Write in your notes that catalysts are chemicals, and we know they're proteins, that reduce activation energy, and that enzymes are biological catalysts. Now we can now see what they do by comparing these two graphs, the ones right in front of you right here. I guarantee you'll see this diagram in the AP exam. It's there nearly every year. In fact, there's been a protein synthesis or enzyme catalysis long or short answer question on the last 4 exams. You'll be doing a long writing on those sample questions on Monday or very shortly. So the takeaway here is that enzymes help speed up reactions by lowering the amount of activation energy required to start the reaction. And you'll have to be able to graphically prove that. So these two graphs side by side, labeled correctly, extremely important and helpful when you're trying to explain what it is that enzymes do. Now let's start the process of looking at enzymes in a little more detail. As we mentioned, enzymes are referred to as biological catalysts, but what does that mean? Write in your notes that they are actually proteins in RNA, and that their job is to facilitate all chemical reactions by increasing the rate of reaction without being broken down or used up in the process. That's sort of the textbook definition of a catalyst. Also write that they reduce the amount of activation energy required in the reaction without changing the amount of G or free energy released or required. Remember, G stands for Gibbs free energy. Now, it should be no surprise that enzymes are required for nearly all biological reactions and that they're highly specific. We refer to this as enzyme specificity. Get that in your notes. Sorry I didn't say that before, so quit shaking your head. Quit rolling your eyes. Remember, there are thousands of different enzymes in our cells whose job is the control of all the reactions that our cells perform. Okay, you probably know now, we mentioned earlier that vocabulary, or I refer to it as jargon, is very important in science. So let's expand your enzyme jargon or vocabulary before we get too much deeper into this enzyme specificity. Write in your notes that each enzyme has its own very specific substrate or reactant which binds to that enzyme to create a temporary coupling where we call the enzyme substrate complex. Let's introduce the best example. Lock and a key, which we'll refer to often explaining enzymes and their mechanisms. Write in your notes that to unlock a door, the key goes in the lock for a short period of time in order to do the actual job of unlocking the door, but then comes out again to be used later. In this example, the lock represents the enzyme and the key would represent the substrate. More about that later. Also write that the product is the end result of a reaction. In our lock and key example, it's the unlocking of the door and ultimately opening it. Then there's the active site. Get in your notes that the active site is the enzyme's catalytic site where the substrate actually fits into the enzyme, and you can see it right here. So in our diagram now, here's our enzyme. This would represent the lock. Here's our substrate, which would represent the key. And then the active site is the spot where the key is in the lock. So that's what we refer to now as the active site. All right, so again, this diagram puts this enzyme jargon and vocab together. In this case, let's look at the products first. So the products are over here. Question: what kind of a reaction is shown in this picture? Now remember, in your mind, or even if you're writing it down, remember to use metabolic jargon. If you said a catalysis or catalytic reaction, you'd be correct. As you can see, we're breaking down a polymer made of two monomers into those individual monomers. So here's our polymer, and here's our two individual monomers. Now let's look at how this was accomplished. Notice that the shape of the enzyme binding site, so here's our binding site right here. It's not labeled. If you want to label it, you can. This is our enzyme binding site. And notice that the shape of it and the substrate now match. We call this a complementary shape. Get that in your notes. And also write that because of this complementary shape, the substrate fits into this specific enzyme. This is what we mean by enzyme specificity. So if these were round instead of the shape they are, they would not fit in this site. So that's what we mean by enzyme specificity, is that this substrate only matches the binding site of this enzyme. Once the substrate binds itself to the enzyme, the reaction can start. So that's at this point in time when this binding takes place, then this hydrolysis reaction can start. Okay, that's it for part 2. So take a picture of the notes that you've just written and submit them to Moodle. Also grab that study guide from part 2 and answer the questions and submit those as well. And I'll see you soon in our discussion of enzymes part 3. Bye.